Okay, so skeletal muscle performance. Let's pick back up with chapter 10. So, myoplasticity, excuse me, changes the muscle structure as a result of changes in function related to physical training. Again, core function, core principle. Majority of mature skeletal muscle fiber nuclei are amitotic. They do not undergo mitosis. Satellite cells, there's a small population of unspecialized cells, do retain their mitotic ability and they can help repair an injured skeletal muscle. Myoplasticity, therefore, changes in response to training. They're found within the training within the muscles. They do not involve changing the number of muscle fibers. Precise change that occurs depends on the type of training, as opposed to endurance training versus resistance training. Endurance training, you have a large increase in frequency of motor neuron activation, moderate increases in force production, more repetitions with a lighter weight. Again, this would be the folks who are doing endurance training. They're training to run marathons or swim long distances or climb mountains or whatever. They use more repetitions with a lighter weight. Causes a primarily biochemical changes, most dramatically in type 1 fibers, but it does make changes in the type 2. It increases the oxidative enzymes and mitochondria and their associated proteins. It increases fatigue resistance, makes the muscle more efficient using fatty acids and non-glucose fuels for ATP production, and it increases in blood vessel network supplying the muscles. So all of these things are brought about by more repetition, lighter weight. Moderate exercise in frequency of motor unit activation and a large increase in force production. This is going to be more for strength training. Fewer repetitions, but with a heavier weight. So here you're doing 100 squats with no weight, just your body weight. Here you're doing 50 squats, but you've got a barbell that has 125 pounds on it on your shoulders. This causes primarily anatomical changes, both in the number of myofibrils and the diameter of the muscle fibers. It increases, and that is referred to as hypertrophy. Resistance or strength training. Yes, endurance, resistance, and strength. With hypertrophy comes decreased proportions of mitochondrial proteins and blood supply to the muscle because of the enlargement of the fiber, not because the mitochondria and vessels are actually lost. You have the same number of mitochondria and the same blood supply, but you have a bigger muscle fiber to supply all those things too. And can decrease endurance. Again, when you start working with big weights, you generally don't have the endurance you have working with smaller weights. So a balanced program combining both types of training is recommended for most people. Again, if you're going to run marathons, you want to do one thing. If you want to be a bodybuilder and be Mr. Olympia, you do another thing. If you just want to be a healthy John Q. Public, you do a little bit of both. Disuse leads to anatomical and biochemical changes, including decrease in the number of myofibrils and the size of the fibers themselves, and decrease in the oxidative enzymes. This is called atrophy. It results in the decline of both strength and endurance. This is the old concept you may have heard at some point, use it or lose it. And it's a particular problem for the bedridden and those who have lost the use of their limbs. That people may say, muscle wasting. The clinical term for that is muscle atrophy. And again, here's the beginning muscle before we started exercising. If you do endurance training, you'll notice the size has not changed very much. But we do have these increased number of mitochondria and mitochondrial proteins. We have an increased number of blood vessels. We went from 2 to 3, and we have increased oxidative enzymes. If we do resistance training to get the strength, we have increased numbers of myofibrils. So if you've got 3, 6, 8, now we've got 3, 6, 9, 12. You'll notice they are bigger. In addition to being more of them, they are bigger. You have an increased diameter of the muscle fiber and the myofibril itself. Here's the disuse, or the atrophy. You have decreased numbers of myofibrils. Up here, see again, we had 8. Now we've only got 6. We've only got 1 mitochondria where we had many more than that. And each mitochondria has fewer oxidative enzymes. So this muscle is wasting away. Muscular fatigue is the inability to maintain a given level of intensity during activity caused by multiple different factors. Depletion of key metabolites, creatine, phosphate, glycogen, and glucose. Decreased availability of oxygen to the muscle fibers. The increased demand, depletion, or depleted myoglobulin-bound oxygen, inadequate intake. Accumulation of certain chemicals in the fiber, the calcium, the ADP, and independent phosphates. Environmental conditions. If it's extremely hot, sweating may cause you to lose those electrolytes. Again, I told you when I was doing 2-a-days in football back in the 70s, they gave us those god-awful salt tablets to replace what we were sweating out. Well, we were sweating out a lot of stuff. All we were replacing was the salt. Post-exercise oxygen consumption and the recovery period. It takes time after exercising to return to a pre-exercise state, which is called the recovery period. You have an increased rate of breathing, which supplies that necessary oxygen. This is called excessive post-exercise oxygen consumption. That allows the body to recover from exercise-induced homeostatic imbalances. Plays a role in heat dissipation, restoration of intracellular and extracellular ion concentrations, and blood imbalances. All right. That wraps up the skeletal muscle. We're going to spend the rest of this chapter talking about smooth and cardiac muscle. And this is going to be sort of a real quick overview. We'll talk more about the smooth muscles when we talk about organ systems, such as the esophagus and the stomach and some other things. And then we'll talk about cardiac muscle when we do the heart. So, smooth muscle propels material through the hollow organs. That is a process called peristalsis. It forms sphincter muscles in the digestive and urinary system, which control passage of materials by opening and closing, and regulates flow rates through the hollow organs like the blood vessels, the respiratory tract, and the gastrointestinal tract by changing the diameter of the tubing. The muscles contract, the tube gets smaller, the muscles relax, and the tube gets bigger. Smooth muscle cells contain myosin and actin fibers arranged very differently than those in skeletal and cardiac muscle. There are no sarcomeres. Therefore, there are no striations. Actin filaments are arranged obliquely, that means at an angle, in the sarcoplasm, and they are anchored to proteins called the dense bodies. Dense bodies in the sarcoplasm are bound to a scaffold-like intermediate filaments, which connect the dense bodies to each other. Other dense bodies link smooth muscle cells to the surrounding muscle cells, which allow tension transmission from cell to cell. You have several thin layers of filaments that radiate from each dense body to surround a single thick filament. The ratio of thin to thick is therefore higher than in skeletal muscle. Both the thick and thin are longer. The thin lacks troponin. The myosin heads are found along the entire length of the thick filament. Think back to the thick filament of the skeletal muscle. Those heads were only found on the ends. Here they're found on the entire length. They have opposite facing heads on either side of the filament. In other words, they're on, instead of all being pointing this way, they point this way. And smooth muscle cells lack motor end plates. Sarcoplasmic reticulum is much less extensive, and there are no T tubules. So there's smooth muscle right there. This is an area of the small intestine. We're cutting away this little section right here. We've blown it up. There's the muscle fiber. There's the intermediate filaments, the ones in green. There's the nucleus, again, spindle-shaped fiber with a centrally located nucleus. There are the myofilaments and the dense bodies. You see the little blue dense bodies, which you see at the end of each myo or along the myofilaments. Smooth muscle contraction and relaxation. Smooth muscle involves different cascading events. You have an influx of extracellular calcium that binds to the protein in the sarcoplasm called calmodulin. The complex activates the myosin light chain kinase, or MLCK. The myosin light chain kinase, in turn, activates the myosin ATPase. And remember, any kind of word ends in ACE, we're talking about an enzyme. And then the cross-bridge cycle begins. Repeated contraction cycles pull actin along the myosin. The cell changes shape from thin to flat to fat and globular. From thin and fat to fat and globular. I'll get that out in a minute. Smooth muscles can contract up to 80% of their resting length, whereas skeletal muscle only contract about 30 to 40% of their resting length. And only about 1/100th amount of ACP is required when compared to skeletal muscle. So even though they contract much more, they require much less energy to do so. The relaxation phase occurs when calcium is removed from the cytosol, the MLCK is deactivated, and the myosin ATPase is deactivated. The latch state is an alternative to the relaxation state that we talked about in the skeletal muscle. This is where the cell remains contracted. However, it is in an energy efficient mode. This is very important in our sphincter muscles. They must stay contracted to remain closed, but they're doing so and they're not burning a lot of energy to do it. If you contract that muscle, you are constantly using energy to keep it contracted. The sphincter muscles are not doing that. So we got smooth muscle here. Here is the resting state of the smooth muscle. You see the myosin heads opposite of each other, not all pointed the same direction as in the skeletal muscle. And then when they get ready to contract, these all attach and pull this way. These all contract, attach, and pull that way. So you take a muscle that did look like this, now looks like this. So thin and flat, fat, thin and flat, fat and globular. Single smooth muscle unit is the predominant type found in the body. It's found in nearly all the hollow organ walls, including the uterus. Linked electrically by gap junctions. The action potential spreads rapidly through the cells via these gap junctions, and the cells contract in a coordinated way as a single unit. Cells respond to multiple stimuli, such as mechanical, hormonal, neural, and local pacemaker cell stimuli if you happen to have a pacemaker. Multi-unit smooth muscles are less common than the single unit type. They generally have individual cells not joined by gap junctions. They contract independently to allow for precision. The amount of tension varies with the number of cells activated and responds primarily to nerve stimulation. Think of the muscles that control the size of the pupil of your eye. These are these multi-unit smooth muscles. Cardiac muscle, structurally similar to skeletal muscle fibers with some major differences. They are shorter, they have branches, and there are 1 or 2 nuclei and a very abundant amount of myoglobin. The mitochondria account for about 30% of the cytoplasmic volume. The areas where the cells are linked together are structures called intercalated discs. They are connected electrically by gap junctions and physically by desmosomes. And all of this construction allows the heart to contract as a coordinated unit. Unlike skeletal muscles, cardiac fibers do not require stimulation from the nervous system to generate action potentials. Electrical activity is coordinated by pacemaker cells. Cardiac pacemaker cells in specific regions of the heart. Autorhythmic, they act like a single unit smooth muscle. They spontaneously generate action potentials. They coordinate the electrical activity and trigger contractions of the cell. Oops. It wraps up chapter 10 and muscle tissue anatomy and muscle tissue physiology. Next time we get together, we will start talking about the nervous system, and we will spend the rest of the semester talking about nervous tissue and then the varying nervous systems. It's Friday afternoon where I am. Y'all have a great weekend. Watch a lot of college football. It's supposed to be fairly pretty Saturday and Sunday, so don't spend your whole weekend out watching football. Get outside. If you have kids, play with your kids, and I will see y'all next week. Have a great day.